So I'd like to invite our next speaker up, Dr. Milka, who specializes in early stage of evolution and implementation of novel NGS and single-cell omics. Received his master's in 2013 at National University of Kyiv and his PhD in 2018 at the University of Ghent for nuclear receptors and their role in inflammation, autophagy, and circadian rhythm. He joined the TechWatch team at VIB in Belgium, where he works on the evaluation and optimization of disruptive single-cell omic technologies to bridge the gap between the tech providers and the researchers. So today he's going to present his work using RNA-seq to analyze single cells.
Thank you very much for introduction and giving this opportunity to present our work. And thank you, of course, for coming here today. Yeah, so I would like to start my presentation with a question. So would you like to do single-cell RNA-seq experiments with less cost, less batch effects, and also being able to process several samples as one sample at once? So I assume the answer is obvious, at least it was for us when we started. Okay, yeah, thanks. So our institute started collaboration with Janssen Pharmaceutica, and the aim was basically to join forces to test, implement novel and back then very challenging single-cell technologies. So we framed several projects, several pillars in the collaboration, and today I'm going to present you one of the projects, namely hashing or sample multiplexing for single-cell RNA-seq. So first of all, I will introduce you to the concept of hashing, then I will show you results on benchmarking of different hashing methods applied on human cells, nuclei, including also human PBMCs from COVID clinical studies. Then we will go over application of sample multiplexing on mice tissues, so cells from mice tissues, and finally we will conclude with practical recommendations and also with, of course, with scientists, we will mention limitations. So the approach, or what is hashing, you basically take your cells from different samples, right? And you label each cell with one DNA barcode. In this example, it's achieved by DNA barcoded antibodies. So you can see sample 1 receives DNA hashtag 1 and so on. And then you simply pull those cells in one sample. And then cell encapsulation, library preparation, and then sequencing, everything happens on one sample instead of 3 in this example. And only during the analysis you are able to deconvolute or demultiplex those cells back to their samples of origin using those DNA barcodes, right? And as I already mentioned, this approach allows first diminish batch effects because you work on one sample instead of 3 in parallel, for example. And of course, it allows you to reduce cost because, for example, 10x or any other single-cell RNA-seq reagents, they are relatively expensive if you consider the whole experimental cost. So that's also a nice feature. And you get an extra information, data layer, so hashtag expression. And then you can better predict your doublets, which are not used usually in downstream analysis. So you need to get rid of them. And that's extra layer of information to predict doublets or multiplets. Those are droplets that contain 2 or more cells. And as Davide nicely summarized, there is a variety of protocols, right, always. And then one of points of our study is to navigate you through those methods, to benchmark them, and also just to confirm that you can apply hashing, you can multiplex different samples, even if they are genetically similar. That's important. So I already introduced you to antibody hashing. Then you can have genetic hashing when you just pull samples that are genetically different. They come from different genetic backgrounds. And then again, you can deconvolute them on basis of SNPs. Or you can introduce those genetic differences by transducing cells with some vectors. There is also an approach to directly label your cells with DNA barcodes through click chemistry. And finally, you can label cells with DNA barcoded lipids, the last example. And in our work, we focused on antibody-based, different antibody hashing, and also lipid hashing, because they were the most promising and available when we started this project. And we used genetic hashing as a ground truth. So we wanted to calculate our hashing of antibodies and lipids, and we used as a basis, as a ground truth, genetic hashing, because we always pulled samples coming from different cell lines, different patients, or different mice strains. So antibody hashing, very popular antibodies are from BioLegend. They are so-called TotalSeq antibodies. They exist in 3 formulations. Each of them specific towards particular single-cell technology. For example, TotalSeq-A can be used with any poly-A-based single-cell RNA-seq methods, while TotalSeq-B only with 3' from 10x, and TotalSeq-C only compatible with 5' chemistry from 10x. For example, if you want to use TCR or BCR sequencing, you have to use 5' kit, right? And then this kit is compatible with TotalSeq antibodies. But in essence, all these antibodies, they target same antigens on human or mice cells. So they are, in fact, biologically the same, just different DNA barcodes. Which lipids did we test? We used MULTI-seq lipid-modified oligos for cells and cholesterol-modified oligos for nuclei. Both of them coming from authors of the original lipid hashing paper, so McGuinness in Nature Methods. And we also designed our own lipids. So we designed and ordered lipids where we mimic the DNA barcode to keep it the same as in TotalSeq-B antibodies. So to exploit benefits of 10x chemistry, 3' chemistry. So that's, I introduced you to kind of portfolio of tools we used. Experimental design, I already mentioned that we used genetic hashing as a ground truth. So in practice, what does it mean? So for example, we take MCF7 cells, we label them with #1, be it antibody or lipids, right? Then PC3 cells, for example, #2, and so on. And then we simply pull those 4 samples into one, load on 10x, library prep, sequencing, everything on one sample. And then we, of course, repeat this for all type of hashing, different lipids, different antibodies, nuclei, so on and so forth. So we have many data sets. And the data analysis the analysis pipeline was applied the same for all data sets. It's a comparison benchmark paper, the study, right? So we wanted to be fair, and we applied the same pipeline. So in fact, FASTQ files, FASTQ reads were mapped with CellRanger. They generated gene cell matrices, which were used as an input for Seurat. Seurat is very standard, very popular tool to do downstream single-cell RNA-seq and beyond, of course. So for gene expression data, we did pretty standard pipeline. So gene counts were normalized until the cell clustering and UMAP plots. Then hashtag expression data, that's a crucial part in our study. These counts were also normalized, different type of normalization though. And key here is, I will even show you these 3 functions. So that's the multiplexing functions which show you then samples of origin, right? So we always evaluated 3 functions, GMM demux, MULTI-seq demux, which was originally published in lipid hashing paper, and HTO demux was published in antibody hashing paper. And the output of this demultiplexing is in essence a table where each cell receives 1 hashtag if it's a singlet, so singlet 1 cell, and then more than 2 if it's a doublet or multiplet. Then gene data were used by FreeMaxLab. So just to let you know, this is a very handy tool. It doesn't require exact genotyping data, so you can separate your samples from the pool without providing exact genotyping data because it used Bayesian model to separate those samples. And the output is again table. Each cell receives 1 tag, 1 sample. Or if it's a doublet, then it's 2, of course. So the final step is to calculate overlap, right? and we call it overall classification accuracy because we count there both singlets overlap and non-singlets overlap. That's important. Or hashing accuracy. And that's the main metrics you will see in this slide. So finally, now let's have a look on results. So here, I would like to show you comparison of two types of antibodies and two types of lipids. So here what we see is expression, or to correctly name it, level of detection of different hashtags detected by antibodies or lipids. And on this heat map, you can clearly see that there is pattern of 4 clusters always, right? So 4 cell lines, 4 clusters. For example, here, PC3 cells, and then you always have this cluster which expresses or contains more than two tags, hashtags, and these are doublets. Then we cluster cells. You see this UMAP plots for each data set, and the color corresponds to ground truth annotation. So the annotation on basis of SNPs or on basis of genetic difference. And you see that each color nicely colors, I mean, each cluster receives one single color, which means that our ground truth is valid. And then when we apply multiplexing function, we see high concordance. So the color here on the lower row corresponding to hashtag demultiplexing. And there you see, as I mentioned, high concordance, especially for antibodies. So it's like almost 100%. You can never get 1 or 100% because you always have few cells which don't get any hashtag due to some technical reasons. But it's pretty high concordance. It's a bit less for lipid hashing. So lipid hashing is inferior towards antibody hashing on cell lines, but it's still acceptable. And then this is to show you an extra control because someone might wonder, okay, how you sure FreeMaxLet is really ground truth? So we checked few gene markers. For example, estrogen receptor is known marker of MCF7 cells, and indeed cluster of CM7 cells expresses estrogen and not other 3 cell lines. So we know that our ground truth is valid. And this is also a bit of maybe technical detail, but important because it's practical. What happens if you load more cells than what the next recommends? The answer is hashing accuracy drops. So from 96% to 91. Why? Because when you increase number of cells, you exponentially increase number of those called negatives and doublets. And they are not used, so the hashing accuracy drops. So there is always balance between cost efficiency and number of cells you load on your lane of your, for example, 10x instrument or any other instrument, in fact, if we are talking about microfluidics. Okay, nuclei, same cell lines, but this time lipids are cholesterol modified oligos and antibodies only one type. Here I show you 2 technical replicates of this experiment because there were issues with nuclei hashing with antibodies. FreeMaxLet, ground truth annotation, excellent work, 4 clusters for colors, and then hashtag based on a demultiplexing, you see that cholesterol works pretty well, 84% correctly, let's call it correctly demultiplexed, and then antibody hashing around half only, so which is pretty low, and the issue comes from one antibody which failed in both experiments. Nevertheless, you can still fine-tune these relatively bad results by improving your computational pipeline, so function which we use for demultiplexing. This is just for illustration. In the paper, we didn't play with those parameters because, as I said, we wanted to make a fair comparison, right? So you know that what happens in a fair battle, let's say. But for this particular bad data set, we just changed one parameter, positive quantile, so threshold, which defines where is negative, where is singlet, and we improved slightly from 51 to 65. So just a message for you, for your biological projects, you always will play with parameters, for example, of demultiplexing functions to decrease number of negatives and to get number of doublets in your demultiplexing results closely towards the theoretical number of doublets. That can be calculated on basis of some formulas, popular formulas, for example, from Satija group. All right, you might say, well, cell lines, no one, okay, not no one, but people don't work really with cell lines in biological projects. So we also applied hashing on human PBMCs from real clinical studies, COVID study, where in our institute there was a big sequencing study on COVID patients, healthy donors. On top of these cells, we applied also 300 side-seq antibodies to detect 277 cell surface proteins. So you can imagine cells underwent also extra washing steps. And then on top, hashing antibodies, because we wanted to increase throughput, and we pulled 3 patients into one sample. And this is this number here is hashing accuracy, 82 for healthy donors, 84 for COVID, which is lower than on cell lines, right? You remember it was 95 or so. But again, this was a big trial, many samples, cells were not as viable, they were stressed, and importantly, we don't filter here in this study any dying cells. We want to show you fair, again, picture. But of course, for your project, for example, you would just filter out these dying cells, and your hashing accuracy would jump up a bit, like until maybe 0.9. Also important message here, because that's a question also from people, okay, what is specificity of this hashing antibodies toward different cell types? Is there a bias, for example, better in T cells than in monocytes? And the answer is, there is no bias. Hashing accuracy is pretty comparable across major superclusters, we call it. So T cells, where is it? Annotation. T cells, B cells, and monocytes and disease. And kind of a confirmation that when we improve cell viability, when we worked with PBMCs coming from commercial vendors, so we did a small experiment, and cells were less stressed because there were less processing steps, no side-seq antibodies on top, then you can see hashing accuracy was much higher for TotalSeq-B antibodies. It was really, I think, the best in what we get from our study. You cannot get more. And for CellPlex, so CellPlex are lipids, in fact, from 10x. They're proprietary hashing reagents, and it was slightly lower than TotalSeq-B antibodies. Nevertheless, again, hashing accuracy was comparable across different cell types. Numbers are not visible, but you trust me, it's more or less the same. Here is a summary of all data from human samples. Here we depicted all 3 functions which we use for the multiplexing. So we were comparing those 3 functions. So you can see in yellow this MULTI-seq MUX, which was the best in our study. And again, antibodies on cells, excellent. Antibodies on PBMCs, also very good. And on nuclei, antibodies are not as great, you can see here, but cholesterol-modified oligos were better than antibodies. So that's the conclusion for the human part. And of course, again, might be a question, well, cell lines, PBMCs, still suspension cells. You don't need to homogenize tissue, so you don't destroy, you don't damage cell surface. So what happens if you have those situations? Because when you isolate cells from tissues, you do homogenization, sometimes enzymatic digestion, right? And we did this benchmark. So we worked with 4 different mice strains. From each mice strain, we isolated 1 type of tissue, brain, lung, spleen, and skin. And we compared TotalSeq-B antibodies and 2 types of lipids. What we learned already from this experiment was that the FreeMaxLab software, so you remember this software which does the multiplexing on the basis of SNPs, it failed to separate black 6J mice with black 6N mice. So they are closely related, and the tool failed because other mice strains were very different, so it couldn't properly annotate those. Nevertheless, we could still draw very useful conclusions from this experiment. Yeah, first of all, we realized that majority of cells were coming from lung and spleen. They were relatively easier tissues than brain or skin. Skin was actually almost, we didn't collect anything. It simply almost failed. But still, the conclusion here is that antibodies work better on lung and spleen cells. So again, they are still processed with digestion, with enzymatic digestion, but still antibodies worked pretty well. The numbers here are, I see 85 or 87% no, sorry, 88% for lung and spleen on antibodies, and lipids work better on brain cells. And brain cells, I'm talking about mostly immune cells, because that's what we've seen when we annotated cells. Then we repeated this experiment. Now we took really 3 different mice strains to make sure we can separate them and calculate ground truth. And we worked only with brain cells, again, immune cells. And conclusion was the same. Lipid hashes work better than antibodies hashes. But still, 73, it's not as good as on human cells or PBMCs. You remember, it was 85, 95, which is a sign of limitations. If you work with complex tissue like brain and homogenization, collagenase, enzymatic digestion, you will not get good hashing accuracy apparently because cells will be stressed. And when you do hashing, you wash, right? You centrifuge to get rid of background. So that's where these sample multiplexing using hashtag reagents suffers a bit. Still, all microglia cells could be demultiplexed. When using lipids, microglia, I mean, we checked few markers, but this cluster, you see, all negatives, which means the demultiplexing function failed to separate them. It was ependymal cells, as our neurobiologist annotated them. And then again, I will remind that we worked with 3 different algorithms. So GMM-DeMux, HTO-DeMux, and MULTI-seq-DeMux. And on average, across all data sets, the best was MULTI-seq-DeMux. And there we used for all data sets, auto threshold equals true parameter. That's maybe an important remark because default is false. But anyway, it was fair because we applied it for all data sets. So the conclusion in this plot, again, on basis of our studies, on our basis of our tissues, if you work, for example, with human cell lines, PBMCs, mice, spleen, and lung, you can use antibody hashing. I'm pretty confident to recommend it. Issues start if you work with brain cells. There, lipids were better, but still not as great. So maybe hashing using these reagents is not the best option. Better to pay for more reagents and do more work, but to capture everything. And on nuclei, cholesterol-modified oligos were better than antibodies. You can read more details on in our paper, more discussions, more details. And that was my last slide. Here is the same conclusion written, but I already mentioned, so I will skip it. And very important, of course, slide to thank all people who contributed to this project, especially group of Professor Ivan Sais. He's professor in Ghent University and also in VAB. And I was affiliated with his group during this project. Also, we worked in collaboration with other centers in VAB, so it was really multi-center effort. So group of Steinarts, Suresh also helped me a lot, and also our colleagues from Janssen contributed to this project. So thank you to them, and of course, thank you very much for your attention.
So we are now pretty much back on track, just a little bit behind. If we've got any questions from anyone in the audience? No, I think that was a fantastic description of the method and running through all of the technical aspects. It was absolutely fantastic. So thank you so much for coming.